Hello everyone and welcome to Facts, Fiction and Stories. Today we are going to learn about cellular respiration, which is the process by which cells convert sugars into energy. This is a very important topic for biology because it explains how living organisms get the energy they need to survive and perform various functions. Before we start, make sure you subscribe to my channel and hit the bell icon to get notified of new videos. Also, if you find this video helpful, please give it a thumbs up and share it with your friends. So, what is cellular respiration? According to Wikipedia, cellular respiration is the process by which biological fuels are oxidized in the presence of an inorganic electron acceptor such as oxygen to drive the bulk production of adenosine triphosphate, ATP, which contains energy. ATP is like the currency of energy in cells. It can be used to power different reactions and processes that require energy, such as biosynthesis, locomotion, or transportation of molecules across cell membranes. But how do cells make ATP from sugars? Well, there are different types of cellular respiration, depending on the availability of oxygen and the type of organism. The most common and efficient type is aerobic respiration, which uses oxygen as the final electron acceptor. Aerobic respiration occurs in eukaryotes such as animals and plants, and some prokaryotes such as bacteria. Aerobic respiration has four main steps: glycolysis, pyruvate oxidation, the citric acid cycle, and the electron transport chain. Let's take a look at each step in detail. Glycolysis is the first step of cellular respiration, and it happens in the cytoplasm of the cell. In glycolysis, a sugar molecule such as glucose is split in half, generating 2 molecules of ATP and 2 molecules of pyruvate. Pyruvate is a 3-carbon compound that can be further processed in different ways. Pyruvate oxidation is the second step of cellular respiration, and it happens in the mitochondria of the cell. In pyruvate oxidation, each pyruvate molecule is converted into a 2-carbon compound called acetyl-CoA and a molecule of carbon dioxide. Acetyl-CoA is then ready to enter the next step, which is the citric acid cycle. The citric acid cycle is also known as the Krebs cycle or the tricarboxylic acid cycle. It happens in the matrix of the mitochondria. In this cycle, acetyl-CoA combines with a 4-carbon compound called oxaloacetate to form a 6-carbon compound called citrate. Citrate then goes through a series of reactions that release 2 molecules of carbon dioxide and produce 1 molecule of ATP, 3 molecules of NAD, and 1 molecule of FAD2 per acetyl-CoA. NAD and FAD2 are electron carriers that store energy for later use. Oxaloacetate is regenerated at the end of the cycle to start again with another acetyl-CoA. The electron transport chain is the final and most productive step of cellular respiration. It happens in the inner membrane of the mitochondria. In this step, NAD and FAD2 donate their electrons to a series of proteins called complexes that are embedded in the membrane. These complexes use the energy from the electrons to pump protons, hydrogen ions from the matrix to the intermembrane space, creating a gradient across the membrane. This gradient drives a protein called ATP synthase to make ATP from ADP and a phosphate group. The electrons eventually reach oxygen, which combines with protons to form water as a waste product. The equation for aerobic respiration shows glucose being combined with oxygen and ADP to produce carbon dioxide, water, and ATP. C6H12O6 glucose plus 6O2 plus 36 ADP, depleted ATP plus 36 pi, phosphate group 6CO2 plus 6H2O plus 36 ATP. You can see that once glucose is completely broken down, the carbon molecules are exhaled as 6 molecules of carbon dioxide, and the oxygen molecules are reduced to 6 molecules of water. The energy released is used to make 36 molecules of ATP per glucose molecule. Biology textbooks often state that 38 ATP molecules can be made per oxidized glucose molecule during cellular respiration, 2 from glycolysis, 2 from the Krebs cycle, and about 34 from the electron transport system. However, this number may vary depending on the efficiency of transport and coupling between steps. Aerobic respiration is very efficient because it uses oxygen, which is the most powerful electron acceptor available in nature. However, it also means that organisms that use aerobic respiration require a constant supply of oxygen, or they will be unable to obtain energy to stay alive. What if there is no oxygen available? Well, some organisms can use other forms of respiration, which are somewhat less efficient. These are called anaerobic respiration or fermentation. Anaerobic respiration occurs in some prokaryotes, such as bacteria and archaea, that live in environments where oxygen is scarce or absent. They use different electron acceptors, such as nitrate, sulfate, or carbon dioxide, to complete the process of cellular respiration. Fermentation is a type of anaerobic respiration that occurs in some eukaryotes such as yeast and muscle cells when oxygen is low or absent. Fermentation does not involve the citric acid cycle or the electron transport chain. Instead, it relies on glycolysis to produce ATP and regenerate NAD+ from NAD. However, glycolysis also produces pyruvate, which needs to be converted into something else to keep the cycle going. There are two main types of fermentation: lactic acid fermentation and alcoholic fermentation. In lactic acid fermentation, one molecule of glucose is broken down into two molecules of lactic acid. The chemical energy that was stored in the broken glucose bonds is moved into bonds between ADP and a phosphate group. C6H12O6 glucose plus 2 ADP, depleted ATP plus 2 pi phosphate groups 2CH3 chaku, lactic acid plus 2 ATP. Lactic acid fermentation occurs in some bacteria and in animal muscle cells when they are working hard and oxygen is low. Lactic acid accumulates in the muscles and causes fatigue and soreness. In alcoholic fermentation, one molecule of glucose is broken down into two molecules of ethyl alcohol and two molecules of carbon dioxide. The chemical energy that was stored in the broken glucose bonds is moved into bonds between ADP and a phosphate group. C6H12O6 glucose plus 2 ADP, depleted ATP plus 2 pi phosphate groups 2C2H5O, ethyl alcohol plus 2CO2 plus 2 ATP. Alcoholic fermentation occurs in some bacteria and in yeast cells. Yeast cells are very good at this form of fermentation and are used by brewers and distillers to create alcoholic beverages. Fermentation is less efficient than aerobic respiration because it only produces 2 ATP per glucose molecule compared to 36 or 38 ATP from aerobic respiration. However, it allows some organisms to survive and produce energy when oxygen is not available. So, that's it for today's video. I hope you learned something new about cellular respiration and how it works. If you have any questions or comments, please leave them below and I will try to answer them as soon as possible. Thank you for watching and see you next time.